So one other kind of tissue system that we'll spend a lot of time talking about kind of has one of the most specialized functions for plants, the very important job of conducting water. So up from the roots into the other parts of the plant, conducting water, and then also conducting sugar away from where photosynthesis happens. So we saw this before, similar image, a cross section of a stem, got these vascular bundles. And then again, the most obvious cell type are these xylem cells. They're bigger, thick walls that stain red. And the phloem cells are more difficult to identify as such. And I often would just identify them based on their position. So toward the outside of the stem from the xylem cells are the phloem cells. And then this zone here, these would be some fibers. So as we'll see, vascular tissue, because it has these thick walls, is also useful for supporting the plant. So it's sort of a pillar of support, these thicker walled, sturdy cells, well placed throughout the stem, keep this plant together. So just a variety of the placements for these. These are showing stems in an early stage of development. So we'll talk about primary growth and secondary growth. So these are the result of primary growth, some of the earliest cells that are produced by a plant. They often have this kind of organization. And then later on, they would develop differently, as we'll see. But you still can tell the presence of the vascular bundles. So yet again, another stem with the bundles of xylem cells. On the left is a dicot. This is alfalfa. And then on the right is a monocot, a corn plant, maize, but also vascular bundles. Just a little bit difference in the way those bundles are arranged, but still xylem and phloem together in bundles. They have kind of a complementary function. So it's important to have the xylem next to the phloem. In the root, again, bundles. This is a primary growth root of a buttercup with xylem and phloem only in the very center. That's a dicot feature. And then on the right, this monocot, the maize plant again, has vascular bundles all throughout in a ring around the outside of this central cylinder. And then leaves, what you see as veins on leaves, those are the vascular tissue. And if you think about leaf veins, they also have a function not only in transporting water and nutrients, but also in supporting the leaf. So those veins are sometimes the more rigid parts of the structure. So again, we've got some fibers making up these bundles and supporting the leaf. So the mid vein of a leaf does a lot of work to support that leaf. If it grows out sideways, parallel to the ground, then it's got to support the weight of the leaf and keep it from collapsing. And that structural support is one of the functions, sort of a side function of these vascular bundles, and then also distributing water to the leaves. So how they look in various sections, xylem and phloem, so in these bundles, basically cells that are elongated is one of the features of these vascular cells. They kind of become like pipes. So you take a sort of like a more of a cube shaped starting cell and stretch it out into, in some cases, a larger diameter cell, but also a long cell. It's got a lot of open space in the middle for transporting the water and transporting the nutrients. So we'll see the functions of xylem and phloem as we have a whole lecture devoted to water and nutrient transport. But again, they exist close together. They have rather different structures and different functions that are complementary. So xylem cells have these thickenings in them. These are circular thickenings or spiral thickenings, and that keeps them stiff. And the phloem cells are a little more tender and juicy. So the various functions here, xylem has the function of transporting water and things that are dissolved in the water up from the roots to the part of the plant that's doing photosynthesis. And the phloem has the job of transporting one of the main things we think about with phloem is transporting the sugar away from the site of photosynthesis. So photosynthesis needs water, which comes up from the roots and produces sugar, which has to go to the rest of the plant. So the whole plant needs this chemical energy of sugar, especially something like the roots that don't have access to the sunlight. So some features of xylem and phloem, I mentioned the thickenings here. So spiral thickenings, also just thicker walls in general. Xylem cells are empty on the inside. They're actually dead when they are mature. So they don't have any cytoplasm, no organelles, no nucleus. They die in place and they become really very much like plumbing. The phloem, on the other hand, does have cytoplasm, and that's important for being able to control the direction of transport and how much sugar is dissolved into these, as we will see a little bit later. So as a process of development, cells that become xylem start alive and they die during that process. So while they're alive, they put together all the structures. So they thicken their cell wall, they put in the chemicals that keep the wall stiff and waterproof. And then they give up on the cytoplasm. And the absence of cytoplasm is convenient for water transport because it kind of would be in the way otherwise to have a nucleus and organelles and all that inside of the cells. If they're dead on the inside, then everything just flows through them. So important feature of the xylem there. Lots of different variety in the way that xylem cells can be put together. So sometimes they're, this just sort of highlights the gamut of sizes and shapes. So short and fat, all the way up to long and skinny. These ones that have open ends are we call vessel elements. The ones that taper more towards the ends are called tracheids. And I'm not going to split hairs too much on those, the difference between those, but you can see them a little bit better in this illustration. Kind of your typical tracheids, your typical vessel elements. Notice they've got sometimes more of an open end, so fully open end or an end with a perforation plate on it. And the tracheids are, they taper towards the end, and so they're more open throughout, but all of these have these pits along their length. So it's not quite like a pipe of plumbing. There's a whole system of lots of vessel elements and tracheids working together. They kind of share water in between their neighbors, but ultimately the job gets done. I'll say that much. And then one other component of the xylem, sometimes they're cells that are just fibers. They don't actually conduct water, but they can contribute to the stiffness of a plant. So this stiffness I've been talking about with xylem cells is in their secondary cell wall, and it's enabled by a molecule called lignin. Lignin is really a category of molecules. So I have a molecule diagram up here. This is like one kind of lignin. It's just such a complex molecule that it doesn't have a strict chemical formula. Again, lignin is kind of a category, but they kind of look like this, and they're put together with these phenolic carbon rings in them, which is part of why they have this chemical, the chemical features that they do. So they're waterproof. Lignin's waterproof, it's stiff, and it's really resistant to decay or breakdown. So a lot of organisms are unable to break down lignin, which makes it very sturdy for the life of the plant, but also even after the plant is dead. Lignin can be very tough to break down. So some examples showing various cells with lignin included in them. Lignin is the reason that these cells stain red. So it's very convenient to look for the presence of lignin by adding this stain and seeing where it goes. So here we've got some of those kind of spiral thickenings. So this is a xylem cell conducting water this way, where the air, where the highlighter is going, and then around its circumference, around that cylinder, are all these thickenings that keep it stiff and supportive. Here's some fibers that have just been sort of smushed up in various directions. Vascular cells, so xylem, water conducting cells, big ones here for big water conduction, also some that are smaller. Uh, like I said, a big variety of sizes and shapes for those, even the ones that are conducting water. Inside of a pear, you can have stone cells, so just cells that have thickened up and basically aren't functioning as cells. They're probably dead as well, with really thick cell walls with lignin in them as a deterrent to being chewed on. And then here's one finally, leaf of a water plant. This is a water lily leaf, and it has this structure in here called an astrosclerid. So astro for star. This is a star-shaped tough thing that helps to keep the structure of this plant in a space where it has spongy airspace tissue. So those astrosclerids help to keep that space open, keep it from collapsing. So if any part of the plant needs stiffness, lignin could be the molecule for you. Lignin also comes up in a kind of a chemical context. So we talked about cellulose and the fact that cellulose is a big component of paper. And what I find kind of interesting that we have wood as the source of our paper. Wood has a whole bunch of lignin in it, and lignin is not super great for paper, right? So we don't need this wood thickness, not as thick as wood really, to be putting our paper together. We need a kind of a delicate, fluffy woodness or a cellulose feature more than a lignin feature. So in the process of making paper from wood pulp, I suppose it's the kind of thing you might have had a lot of excess of. So take our fragmented bits of wood and turn those into paper. During the process, the lignin can be removed. So somewhere on here is, there we go, oxygen delignification. So there's a step in paper production that's removing lignin, bleaching. So sort of more like brown wood becomes bright white paper. In this video, if you want to get it from the links, shows a person who is taking chunks of wood and removing the lignin from them, and then basically turning them into transparent things that used to be wood. Every once in a while, you see people who are trying to kind of invent a new structural use for wood. It's sort of like a natural plastic, if you think about it. And then some of these processes make it so it doesn't really look like wood anymore. So it could be used for a variety of things if the price is right, I suppose. This one was from not too long ago, a study from UW-Madison that was taking lignin from wood, which again is pretty abundant, and using that in a chemical process to make acetaminophen, which is a painkiller. So like the headline here, relieving 2 headaches with 1 process. So this is another version of the lignin molecule, but basically these phenolic rings under the right chemical conditions can be modified into something like this. Don't know what happened of this study. Maybe that's the new source of acetaminophen. And then also lignin features importantly in the fossil record. So these things that we have as fossil fuels, we kind of have the lignin and the durability of wood. We have that to thank for the fact that these plant structures stuck around for so long. So this rather chemically complex lignin molecule was part of the wood of the trees that fell and became today's coal. We talked before about the Carboniferous period. So this period in the past of when a lot of the coal was established, that was there because these trees fell in a location and they did not decompose, and instead they got pressurized and modified into today's coal. So maybe it wouldn't have had the same effect if they were gooey, juicy, herbaceous plants, but thick, woody plants, a little bit better chance of surviving of those molecules sticking around over this time. Okay, so a lot about xylem, now a little bit about phloem. As I mentioned, these phloem cells are also alive, or sorry, in contrast, they are alive at maturity. So we start with a juicy cell and modify it. It's alive a little bit. So it's also still kind of modified for transport. but it's transporting sugars and other molecules inside of cytoplasm. So it's not as like pure water, not quite the same as xylem. So they're sort of modified to have a streamlined central transport. The ends of an individual phloem cell are these pores, these sieve pores. And what's not shown on this diagram are that phloem cells have what we call companion cells. So the final cell division that makes a phloem, one of these open sieve member cells, the final division makes one of those, but also keeps a companion cell, which is a more complete cell that has nucleus, organelles, and so on. And the companion cell is important for sort of loading and unloading that cell. So they're joined by their shared cytoplasm, and this companion cell, if it's in a leaf cell, or if it's in a leaf, I should say, would be putting sugar into the sieve tube. If it's in a root, it might be taking the sugar out of the sieve tube. And so the plant has some control over what goes in and out of the phloem cells. All right, so those are our cell types, and then we'll start putting them together in the growth of a plant.